Welcome to Anatomy and Physiology 2 lecture series. And the book we are going to be covering this semester is a book by Michael McKinley, Anatomy and Physiology: An Integrative Approach. The first chapter in this course is Chapter 17, The Endocrine System. This chapter divided into 3 parts. In this first part, we will discuss and understand how the endocrine system works, and we will cover the following: some basic terminology, relation between the control system, nervous system, and endocrine system, general functions of the endocrine system, location of endocrine glands, types of stimulation of endocrine glands, types of hormones produced and secreted by glands, hormone transportation methods, interaction between hormones and target cells, control systems of the body. Endocrine and nervous systems are the 2 control systems of the body. They interconnected and work together to regulate bodily functions and maintain homeostasis. Both the endocrine and nervous system release ligands, chemical messengers. Ligands bind to cellular receptor on particular target cells. The nervous system uses 2 types of intercellular communication: electrical and chemical signaling, either by the direct action of an electrical potential, or in the latter case, through the action of chemical neurotransmitters, such as serotonin or norepinephrine. Neurotransmitters act locally and rapidly. When an electrical signal in the form of an action potential arrives at the synaptic terminal, they diffuse across the synaptic cleft, the gap between a sending neuron and a receiving neuron or muscle cell. Once the neurotransmitters bind with receptors on the receiving postsynaptic cell, the receptor stimulation is transduced into a response, such as continued electrical signaling or modification of cellular response. The target cell responds within milliseconds of receiving the chemical message. This response then ceases very quickly once the neural signaling ends. In this way, neural communication enables body functions that involve quick, brief actions, such as movement, sensation, and cognition. Endocrine signaling. In contrast, the endocrine system uses just one method of communication: chemical signaling. These signals are sent by the endocrine organs, which secrete chemicals, the hormone, into the extracellular fluid. Hormones are transported primarily via the bloodstream throughout the body, where they bind to receptors on target cells. As a result, endocrine signaling requires more time than neural signaling to prompt a response in target cells. The amount of time varies with different hormones. For example, the hormones released when you are confronted with a dangerous or frightening situation, called the fight or flight response, occur by the release of adrenal hormones, epinephrine and norepinephrine, within seconds. In contrast, it may take up to 48 hours for target cells to respond to certain reproductive hormones. Then we will discuss a detailed comparison between the nervous and endocrine control systems, highlighting their major differences in terms of functionality and characteristics. 1. Signal type. The nervous system utilizes electrical impulses and neurotransmitters to transmit signals, whereas the endocrine system relies on chemical messengers known as hormones. 2. Speed. The nervous system operates at a very fast pace, typically in milliseconds. In contrast to the endocrine system, which acts more slowly, with effects ranging from seconds to several days. 3. Duration of effect. Effects initiated by the nervous system are generally short-lived. On the other hand, the hormonal effects induced by the endocrine system tend to be long-lasting. 4. Target specificity. The nervous system is highly specific in its targeting. Signals are directed precisely to particular neurons or muscles. Conversely, The endocrine system has a broader reach as hormones travel through the bloodstream and affect multiple cells. Pathway. In terms of pathway structure, the nervous system uses neurons, nerve fibers, for signal transmission, while the endocrine system utilizes the bloodstream for hormone distribution. Control center. Central control within these systems also differs. The brain and spinal cord, central nervous system, govern neural activities, whereas hormonal regulation is managed by the hypothalamus-pituitary axis along with other endocrine glands. Examples. Practical examples include reflexes, muscle contractions, and sensory processing within the nervous system. In contrast, growth processes, metabolic regulation, reproduction functions, and stress responses are typical activities governed by hormones in the endocrine network. How they work together. There are examples in which the nervous controls the endocrine system, like when the CNS, hypothalamus, to control the human behavior, including emotional and stress responses, or basic drives such as sleep, hunger, thirst, and libido. Also, hypothalamus regulates pituitary secretion by hormonal substances carried in the hypophyseal portal blood vessels. Other examples in which the hormones influence the nervous system. For example, reproductive hormones affect brain development and function. Testosterone was related to toughness and leadership behaviors in adolescent boys. Both systems work to maintain homeostasis, ensuring the body's internal environment remains stable. So the works in coordination example of this when the hormones help the body to respond to stress, the adrenal glands in response to stress by releasing EP and NP. Pathways by which the nervous system influences hormone secretion by hypothalamic pathway through hypothalamus or direct neural stimulation through autonomic nervous system. The hypothalamic pathway. This done through the hypothalamic pituitary axis. The hypothalamus, located in the brain, receives neural signals from other brain regions and translates them into chemical signals in the form of regulatory hormones, releasing and inhibiting hormones. These hypothalamic hormones are transported to the anterior pituitary. Then they stimulate or inhibit the secretion of specific hormones from the anterior pituitary gland. The hypothalamus also directly controls the posterior pituitary. Hormones like oxytocin and antidiuretic hormones, ADH, are produced by neurons in the hypothalamus and transported down their axons to the posterior pituitary, where they are stored and released into the bloodstream in response to neural stimuli. The direct neural stimulation. The nervous system can directly stimulate the adrenal medulla. A classic example is the fight or flight response, where the sympathetic nervous system directly stimulates the adrenal medulla to release epinephrine and norepinephrine, adrenaline and noradrenaline. These hormones produce widespread physiological effects like increased heart rate, dilated blood vessels, and suppressed digestion and immunity, preparing the body for action. The nervous system can directly stimulate endocrine glands to release hormones through the autonomic nervous system, like the secretion of gastrointestinal hormones. What are the general functions of the endocrine system? Regulating development, growth, and metabolism. Hormones help regulate embryonic cell division and differentiation. Hormones regulate metabolism, both anabolism and catabolism, maintaining homeostasis of blood composition and volume. Hormones regulate blood solute concentrations, for example, glucose, ions. Hormones regulate blood volume, cellular concentration, and platelet number. Controlling digestive processes. Hormones influence secretory processes and movement of materials in digestive tract. Controlling reproductive activities. Hormones affect the development and function of reproductive systems and the expression of sexual behaviors. Basic terminology. The endocrine system consists of cells, tissues, and organs that secrete hormones as a primary or secondary function. Endocrine organs differentiated into endocrine glands proper, which are ductless glands, secrete their hormones directly into the interstitial fluid and the blood vessels. The blood transports the hormones throughout the body. These include the pituitary, thyroid, parathyroid, adrenal, and pineal glands. Organs that contain endocrine cells like pancreas, hypothalamus, thymus, heart, kidneys, stomach, small intestine, liver, skin, ovaries, placenta, and testes. Types of stimulation of endocrine glands. When high blood glucose levels stimulate the pancreas to release insulin, this called humoral stimulation. Humoral stimuli involve changes in blood levels of ions, nutrients, or other non-hormonal chemicals that trigger hormone release. Hormonal stimulation involve one endocrine gland being stimulated by a hormone released from another endocrine gland. This often occurs in a hierarchical manner, where the hypothalamus releases hormones that stimulate the anterior pituitary gland, which in turn releases hormones that stimulate other endocrine glands. For example, the hypothalamus releases thyrotropin-releasing hormone, TRH, which stimulates the pituitary gland to release thyroid-stimulating hormone, TSH, which then stimulates the thyroid gland to release thyroid hormones. Neural stimulation. Neural stimuli involve direct nerve stimulation of endocrine glands to release hormones. This is typically seen in situations requiring a rapid response, such as the sympathetic nervous system stimulating the adrenal medulla to release epinephrine and norepinephrine in response to stress. Homeostasis of hormone levels. Feedback systems. Hormone levels in the body are maintained through feedback systems, primarily negative feedback loops, which ensure that hormone levels stay within a specific range to maintain homeostasis. These loops involve a stimulus, a response, and a feedback mechanism that either increases or decreases hormone production based on the body's needs. Negative feedback loops. Negative feedback loops work by sensing changes in hormone levels and then triggering responses that counteract those changes. Most of feedback loops are negative feedback systems. Example: when thyroid hormone levels rise too high, the hypothalamus and pituitary gland are signaled to reduce the release of thyroid-stimulating hormone, TSH, which in turn decreases thyroid hormone production, bringing levels back to normal. So, negative feedback loops are crucial for maintaining homeostasis by preventing hormone levels from getting too high or too low. The other feedback mechanism is the positive feedback loops. In contrast to negative feedback, positive feedback loops amplify the initial change, pushing the system further away from its set point. Example: during childbirth, oxytocin release stimulates uterine contractions, which in turn cause more oxytocin release, leading to increasingly strong contractions until the baby is born. Positive feedback loops are less common in hormone regulation and are typically involved in processes that need to be completed quickly and decisively, such as blood clotting or childbirth. The types of hormones produced and secreted by endocrine glands. The hormones of the human body can be divided into three major groups on the basis of their chemical structure. The first category is steroid or steroid-like hormones. Steroid hormones are derived from the lipid cholesterol. For example, The adrenal glands produce the steroid hormone, aldosterone, cortisol, and the reproductive hormones, testosterone, and the estrogens. Biogenic hormones. All of them water-soluble, except thyroid hormones. These are derived from single amino acids, like tyrosine or tryptophan. Examples include epinephrine and melatonin. Proteins and peptides hormones are water-soluble hormones, categorized in 3 groups. Peptide hormones. These are the most common type of water-soluble hormones and are composed of chains of amino acids. Examples include insulin, glucagon, and growth hormone. Protein hormones. These are larger, more complex molecules composed of amino acid chains. Examples include follicle-stimulating hormone and luteinizing hormone. Glycoprotein hormones. These are protein hormones with attached carbohydrate chains. Type of hormones according to mode of secretion or action. Classical endocrine hormones. Hormones secreted into the bloodstream and travel to distant target cells. When a neurotransmitters released by neurons enter the bloodstream, it called neurohormone or endocrine hormones. Local hormones are produced and released by cells or tissues and act on neighboring cells or the same cells that secreted them. When local hormones act on nearby cells without entering the bloodstream, they call paracrine hormones, the most common form of intercellular communication. An example of these hormones, clotting factors. When local hormones affect the same cell that secreted them, they called autocrine hormones. An example of these hormones, interleukin-2, transport of hormones in the blood, water-soluble hormones. These hormones, including proteins and some amine hormones, can dissolve in the blood and travel easily to their target cells. All the hormones are considered as free active hormones, and they metabolize rapidly. For that, they have a very short half-life in the blood, so we cannot take them orally like insulin. Transport of lipid-soluble hormones. Steroid and thyroid hormones are not water-soluble and need carrier proteins to travel through the bloodstream. These carrier proteins, mainly produced in the liver, bind to lipid-soluble hormones, creating a complex that is also water-soluble and can be transported in the blood. So, only a small fraction of a hormone is unbound, free, in the blood at any given time. This free hormone is the form that can bind to receptors on target cells and exert its effects. Remember, the biological half-life of a hormone is the amount of time necessary to reduce the hormone concentration within the blood to 1/2 of what had been secreted originally, or measured previously. Generally, water-soluble hormones have a relatively short biological half-life, which amounts to a few minutes or less for small peptides and about an hour for larger proteins. Steroids generally have the longest biological half-life, since their transport protein protects them from early destruction or loss. The biological half-life of testosterone, for example, is 12 days. Note that the shorter the biological half-life of a hormone, the more frequently it must be replaced to maintain its concentration in the blood. What determines how a target cell responds? Hormones interact only with target cells, cells with receptors for those hormones, to initiate a specific cellular response. A specific hormone generally has different types of target cells. For example, the hormone insulin binds with muscle cells, hepatocytes, liver cells, and adipocytes, adipose connective tissue cells. The greater the number of different target cells, the wider the potential influence that may be exhibited by a given hormone. Target cells, interactions with hormones. The specific process of how hormones interact with cell receptors, then it initiate a subsequent cellular reactions, is done through interaction of receptor with hormone. The message a hormone sends is received by a hormone receptor, which is a protein located either inside the cell, usually for lipid-soluble hormones, or within the cell membrane, and these receptors for water-soluble hormones. The receptor will process the message by initiating other signaling events or cellular mechanisms that result in the target cell's response. Hormone receptors recognize molecules with specific shapes and side groups and respond only to those hormones that are recognized. Mechanism of the intracellular actions of lipid-soluble hormones. Lipid-soluble hormones, such as steroids and calcitriol, are small, non-polar molecules that are lipophilic or lipid-loving. The plasma membrane typically does not pose a significant barrier to these molecules due to their non-polar nature. As a result, the unbound lipid-soluble hormone diffuses readily through the plasma membrane and binds with an intracellular receptor, either within the cytosol or the nucleus, to form a hormone receptor complex. The hormone receptor complex then binds with a specific DNA sequence called a hormone response element. This binding stimulates mRNA synthesis. mRNA exits the nucleus and is translated by a ribosome in the cytosol. A new protein is synthesized. Mechanism of the intracellular actions of water-soluble hormones. Hydrophilic or water-soluble hormones are unable to diffuse through the lipid bilayer of the cell membrane and must therefore pass on their message to a receptor located at the surface of the cell. Except for thyroid hormones, which are lipid-soluble, all amino acid-derived hormones bind to cell membrane receptors that are located, at least in part, on the extracellular surface of the cell membrane. Therefore, they do not directly affect the transcription of target genes, but instead initiate a signaling cascade that is carried out by a molecule called a second messenger. The hormone, the first messenger, binds to a specific receptor on the cell's surface. Since the hormone is water-soluble, it cannot easily cross the cell membrane. Binding to the receptor initiates a signal transduction pathway inside the cell, G protein activation. The receptor is associated with an intracellular component called a G protein, which is activated upon hormone binding. G proteins are a crucial component as they link the receptor to the production of second messengers. Hormone, first messenger, binds to the receptor and induces shape change to activate the receptor. G protein binds to activated receptor. GDP is bumped off and GTP binds to G protein. G protein is then activated. Activated G protein with GTP is released from the receptor and moves along the inside of the plasma membrane, which results in formation or availability of second messenger. Second messenger generation. The first second messenger is activation of budenylate cyclase. The activated G protein then activates an enzyme called adenylyl cyclase, also known as adenylate cyclase. Adenylyl cyclase converts adenosine triphosphate, ATP, to cyclic adenosine monophosphate, CAMP. The second messenger, CAMP, activates a type of enzyme called a protein kinase that resides in the cytosol, specifically protein kinase A, pKa. Activated protein kinases trigger a phosphorylation cascade, meaning they phosphorylate ADF phosphate group to various cellular proteins, including other enzymes. This phosphorylation can activate or inactivate enzymes and other proteins, ultimately altering the cell's activity. The other second messenger is activation of phospholipase C. After hormone, for example, epinephrine binds to its receptor, G protein is activated. Activated G protein activates strong phospholipase C. Phospholipase C splits PIP2 into two second messenger molecules, diacylglycerol DAG and inositol triphosphate IP3. DAG is a second messenger of the membrane that activates protein kinase C. Protein kinase C phosphorylates other molecules. IP3 is a second messenger that leaves the membrane and causes an increase in the levels of calcium in the cytosol. Increase caused by effects on endoplasmic reticulum and cell membrane calcium channel. Calcium acts as a third messenger, activating kinases, sometimes by binding to calmodulin and interacting with ion channels. Target cells are specialized cells that respond to hormones by displaying receptors that circulating hormones can bind to. Despite being transported throughout the body via the bloodstream, hormones only affect a select number of these target cells due to their specific receptor interactions. The activation of target cells is influenced by three main factors. 1. The concentration levels of the hormone in the blood. 2. The relative number of hormone receptors present on the target cell. 3. The affinity between the hormone and its receptor. These factors can be modulated to control how a target cell responds. For example, the presence of a significant level of a hormone circulating in the bloodstream can cause its target cells to decrease their number of receptors for that hormone. This process is called downregulation, and it allows cells to become less reactive to the excessive hormone levels. When the level of a hormone is chronically reduced, target cells engage in upregulation to increase their number of receptors. This process allows cells to be more sensitive to the hormone that is present. Cells can also alter the sensitivity of the receptors themselves to various hormones. Influence exerted by other hormones. Two or more hormones can interact to affect the response of cells in a variety of ways. The three most common types of interaction. The first one is the permissiveness. An example of this is when the thyroid hormones increase the number of receptors available for epinephrine at the latter's target cell, thereby increasing epinephrine's effect at that cell. Without the thyroid hormones, epinephrine would have only a weak effect. Antagonism. The best example of this is the regulation of blood glucose concentration through negative feedback, illustrates how the endocrine system maintains homeostasis by the action of antagonistic hormones. Synergistic. The best example of this action is the effect of testosterone and follicle-stimulating hormones for normal sperm production, spermatogenesis. We hope you found this overview informative and helpful as you learn more about the endocrine system's complexities. If you haven't already, please subscribe